We're going to start with a speaker who needs no introduction, Dr. Jim Wilson, University of Pennsylvania. As you hopefully heard, we recently announced major progress on an ongoing partnership with Dr. Wilson and his team, where the preclinical research we've collaborated on have advanced to a major milestone that supports further advancements of major research into the IND-enabling studies for a first-in-human clinical trial. So we're really getting the research ready for IND studies. We've been working towards this go or no-go decision. Thrilled to announce it's a go. So with that, Dr. Wilson.
Look at my slides. Thank you. Well, it's incredibly exciting to be back again this year with really an important milestone in the evolution of our relationship with FAST, and delighted to share with you what our plans are. And it's really been an interesting year for all of us. A lot of things going on in the world, and some of which has impacted on our on the work that we're trying to do in the laboratories and in biotech and in the pharmaceutical industry. I want to just begin by acknowledging the partnership that has come together. There are many, many, many other people that contribute to this, but now we're sort of, these four are fused at the hip and meeting regularly as a team to try to progress gene therapy for Angelman syndrome. However, over the last year, as was referred to earlier, there have been some troubling developments in diminishing the support for developing treatments, genetic medicines for rare diseases. I don't know if this was a rhetorical question from Goldman Sachs, but they asked, is curing patients a sustainable business model? Well, I guess for the patients or those living with rare diseases, somewhat of a perverse question, but unfortunately it is a question that's being asked now, and the answer from many who are involved in decisions to invest money is it's not a sustainable business model. And this just is the carnage of last year, just a microcosm of the retreat of support for rare diseases, two of which directly impacted on the Angelman syndrome, two of which that we were involved in. My estimate is that gene therapy studies for rare diseases that were funded 2 years ago, 90% of those are no longer funded. It's one of the reasons why organizations like this exist, and it's one of the reasons why your leadership in times like this are so incredibly important. So as I think about why are we struggling with this concept, I think about a business leader, Clay Christensen, who I became a disciple of, who popularized the idea of disruptive technology. Disruptive technology is an innovation that fundamentally changes the way you do business. So for gene editing or gene therapy, this is one-time infusion that lasts for a very, very, very long period of time, as opposed to a pill or an injection that you deliver. And how do you price that? How do you deliver that? How do you assess that? And those are some of the questions that are being asked. Disruptive innovation, however, relates to how can we incorporate those disruptive technology into current market structures. And some of the examples in the past that represent disruptive technologies is how email disrupted fax machines. Probably some of you in the room may not remember fax machines, but a lot of us do. Cars disrupted horse and buggy, and streaming video disrupted movie discs. It turns out that disruptive technologies often begin in niche markets, smaller markets, where proof of concept is developed and therefore expanded into larger markets. And that was our proposal when we brought gene therapy into rare diseases. So we're going to talk now about how we are approaching the development of gene therapy for rare diseases and where does Angelman syndrome fit in. We think it's important to start thinking about this technology not as individual programs but as platforms. And as indicated here, maybe the only difference between one product and another product is the gene that you cassette into the delivery vehicle. So we actually have made a lot of progress with stakeholders, including health authorities across the world, to think about the review and clinical development of products within a platform and what the advantages would be. When you think about a platform, it's important you keep as many things similar or identical as possible, such as the delivery vehicle, which is the vector, the way you deliver the vector, the route of administration, and most importantly, the manufacturing. And if you can be part and participate in a platform as an individual program, I think we can realize some benefit, which is this could result in a much higher probability of success because one program de-risks the other and minimizes the chances for toxicity. We believe it will simplify clinical development, which means faster timelines and less cash. And from a business standpoint, aggregate these programs so that maybe individually they may not be attractive from a market perspective, but collectively they will. So now I just want to talk about what led us to our proposal to work with FAST, and then how this slots into the development of a platform. It really comes down to we know what the gene is, we know what we need to do with the gene, we need to get it to the right cells, which are cells of the central nervous system. The holy grail of drug development for neurologic diseases is broad distribution of that drug throughout the brain. And there are significant barriers for that, especially the delivery vehicles or vectors that are quite large and cannot penetrate barriers. So an intravenous delivery of a gene therapy vector at this point is really not achievable. What the community did several years ago for global neuropathic diseases was somewhat first generation and really has no longer been pursued, but in fact was in clinical trials, which was to deliver the vector into the brain through repeated and multiple direct injections to get a broad distribution of the gene. What we decided to do is to focus more on injecting the vector into the fluid that bathes the spinal cord in the brain, the cerebral spinal fluid. So the approach that we popularized about 10 years ago and has become the dominant strategy for most global neurologic diseases is an injection of vector into the cerebral spinal fluid. Where this is traditionally accessed in the practice of human medicine, it is down around the lumbar space, down around your waist. Turns out in the veterinarian world, the way that veterinarians access the cerebral spinal fluid is below the occipital bone in something called the cisterna magna. And there are reasons why we selected the cisterna magna for the delivery of the vector, and it simply was how to achieve the broadest distribution to the affected organ, which is the brain. What this figure shows is an experiment that was done in which we tagged the vector, injected it into the cisterna magna of a monkey, and then imaged that animal from an hour to 72 hours later. And where it shows up as black is where the vector goes. And you can see it's primarily around the brain, and that correlates to where the gene goes as well. So it's called an ICM, or an intracisterna magna injection. So the platform that we've developed, that we are leveraging for our work with FAST, is illustrated here, and kind of a wonky acronym HIAP, which I'll take credit for or not. But there are three components that we've aggregated in this platform, which is the vector delivery vehicle, and this is a vector that we discovered many years ago called AV9, and we modified into something called HU68. The injection is the cisterna magna, and importantly, the manufacturing has to be consistent, robust, incapable of producing enough vector to conduct clinical trials, but if the product is approved, then supply commercial. And this was a method that was developed based on a platform that's adherent in something called an icelis. And it's pretty much the standard approach that is used in the field. So through the years, we've been evolving this platform in many different diseases, working in parallel with FAST to evaluate preclinical models in terms of demonstrating the potential for efficacy. Our most advanced group of diseases in which HIAP has been developed are called lysosomal storage diseases. I'm going to show you one example where we have 9 diseases, 3 that are in the clinic, all showing promising results. We've also leveraged this approach with a slightly different capsid for an adult form of dementia, which is called frontal temporal dementia. More recently, we've become aware of it because it's the disease that Bruce Willis has, in clinical trials in multiple sites. And interestingly, the same approach can be used for CNS malignancies and neurodevelopmental diseases, and this is where the work with FAST comes in. This same platform can also target cells of the spinal cord, and in preclinical programs, we're addressing some pretty significant ones, including spinal muscular atrophy and ALS. But the reason that I bring this up is when you think about what we're planning to do with FAST and whether you want to be part of it, part of the story that we're going to continue to evolve is all the work on the platform and other diseases that are going to inform potential efficacy and safety in this application. So the injection of vector into the cisterna magna, which is a standard practice in the veterinarian world, was not so much in the human side of medicine. Many decades ago, it was a way in which one accessed the cerebrospinal fluid. But when we proposed this early, I recruited the help of a young interventional radiologist, Brian Pucamus, who really took it upon himself to develop the technique, which requires pre-imaging and then under guidance, introduction of the needle into the cisterna magna. The device is not complicated. It's a syringe and a needle with a tube, small tubing. But we now, through Brian, have trained centers around the world and are involved in basically qualifying all practitioners through on-site tutorials with human cadavers and monkeys to train them in the technique. So the initial application in this approach was in the lysosomal storage diseases. When you do inject the vector into the cerebrospinal fluid in animal models, and we believe also in humans, we do get broad distribution in the brain. You only get access to tissue when you analyze this in preclinical models. But there's broad penetration of the vector throughout the brain, but not every cell apparently is expressing the gene. And it's hard to quantify, but it's less than 100%. And the question is, would we need expression of the protein in all cells to get an effect? And there is a biology in lysosomal storage diseases called cross-correction, and it's illustrated here. Down on the left, we have a vector-treated cell that expresses a protein that can be secreted and taken up by an adjacent cell that's not vector-treated, and that adjacent cell would benefit as much as the cell that received the vector. So this allows you to get a broader sort of bang for your buck based on how much vector that you actually deliver. The most advanced of our programs is a disease called GM1 gangliosidosis. Too many words on this slide, but it's an infantile form of dementia where babies are born normally, but they're noted to be not achieving milestones by 6 months of age, and then basically have a progressive loss of brain in a degenerative disease, which is very different than Angelman, in which then they often succumb to the disease within about a year and a half. This just shows 3 diseases, our 3 lead diseases in a pathway that we have in the clinic, and GM1 is one of them. One of the advantages of these diseases is there are biomarkers that you can measure that are directly related to how defective the gene is, and you can measure them in the blood in the cerebral spinal fluid. So I'll just share with you the clinical data in this approach, because it is the same technology, same route, same manufacturing we're going to use in Angelman. This now finished a phase 1/2 study, and now considering moving into a pivotal study. And what we have here on the top is serial measurements in the cerebral spinal fluid of the protein that's expressed from the gene that we introduced into the brain. And you can see that at baseline, or at the beginning, it was very low, and then it went up, and actually is sustained at levels above normal. And then down below is a biomarker, the accumulation of substrate, that is very high, and then you can see that it reduces to the point where it's approaching normal by 1 year of age. We've tracked the infantile version of this disease in subjects that have been enrolled in the study. They're all surviving, and according to natural history studies, they all would have succumbed to the disease. So we're really excited about this and expect to see similar data with the other members of the platform in the lysosomal storage diseases. So with that as background, and having developed data in generating an Angelman syndrome specific vector, we decided it's probably time to ask the question, should we move into the clinic? And I think with all the background with the platform and the preclinical data generated with support from FAST, the answer was yes. And that's where we're going to So if you think back on this slide about the platform, we're going to use everything that we've used in all those diseases, except we have to swap in the gene. And this is where, through a couple years of work, we optimized the expression of this, and this is the UBE3A isoform 1 gene cassette that we incorporated into our vector from a promoter that is expressed in neurons. We've shown, and others, that in the AS animal model, when injected into the cerebral spinal fluid, that we can show a correction of some of the behavioral and motor abnormalities in this disease. And if you look at the black, is a wild-type vehicle control. Red is an untreated AS mouse, and then a light and dark blue. You see that in the higher dose vector, you can completely correct this abnormality in nest building. And then in terms of rotarod, the same kind of approach. There is the wild-type vehicle. The animals can hang on longer. That's in black. And red is the untreated animals. And then we get a partial correction of this phenotype as well. And there's a whole category of tests that we can deploy against the mouse model to show that there is potential effectiveness. In the drug development world, we call that pharmacology. In terms of safety, we believe the most important species to vet any potential adverse toxicity is a non-human primate, which we do a lot of work in, and I'll just, when I summarize, I'll point that out. By the way, the product that we've identified and declared as a clinical candidate is called GTP220. So hopefully we'll be hearing a lot about GTP220 over the next few years. So we've done experiments where GTP220 is infused into the cerebral spinal fluid, and then the animals are followed over time for any clinical manifestations of toxicity, and then at designated time points, the animals are euthanized and tissues are obtained to evaluate for expression of the gene and any evidence of toxicity, looking at pathology. And we do get a broad distribution of the vector and the expression of the vector throughout the central nervous system of monkeys. And importantly, we have little, if any, evidence of toxicity based on histopathology. And I'm going to comment on that right at the end because we're entering into our formal called GLP toxicity studies. No systemic consequences either. So there's another story that evolved. This isn't our story, but it's surely a kind of interesting and maybe a good story from work supported in other laboratories. And that is it possible that there is cross-correction with UB3A? We don't think this is necessary for us to show an effect based on our current platform, but if it was, it would just extend the reach of our vector and even potentially lead to a broader distribution of correction. This is a pretty aesthetic picture, but what it is, it's a histopathological evaluation of a brain tissue from a mouse that received our vector. And it's analyzed for the expression of RNA in red from the vector, and then protein in green. And in gray, that is the background. Now, if a cell expressed both protein and RNA, it would be purple. So let's talk about what we're seeing here. We see green, gray, red, and purple. What does that mean? Well, if it's gray, there probably was either no DNA vector or the DNA was there and it wasn't expressed well. And that's one thing that we've seen a lot of. And just because we can't detect it doesn't mean it's not there. But in this experiment, that's the readout. If, however, the DNA expresses RNA, it will be red. And if it also expresses protein, is really what you expect it to do, because RNA translates protein, it will be purple. And you do see a lot of purple, which is exactly what we expect. But the interesting aspect of this experiment is the green. Green are cells that have the protein without the RNA. And the only way that that can happen is the cells that did get the gene, express the RNA, and express the protein, are transferring that protein to adjacent cells. So the notion that there may be cross-correction in this disease is really fascinating. Again, we didn't make this observation, but in our vector and our models, there are data to support that as well. So we're now moving into, I'm going to show you a timeline on critical experiments that we're tracking together with FAST. The first is formal pharmacology and toxicology experiments with vector made in the same way that we're going to produce it in the clinic. And these are studies being done at Penn in a FDA GLP certified laboratory. But to possibly provide some assurance, we've done this experiment in 541 monkeys. So this isn't our maiden voyage. And what did we learn from that? Well, we supported many, many other clinical trials in IND-enabling studies that supported moving this technology into the clinic. And the only toxicity that we ever saw was not clinically relevant, but it was evidence for mild inflammation in neurons that, sensory neurons of the spinal cord, they're called dorsal root ganglia. So we've developed a whole strategy around characterizing this and minimizing this. The good news is, in the spectrum of 541 monkeys that we've studied, when we evaluated our pilot monkey studies using, for this program, there was essentially no or trivial levels of any evidence of inflammation in this part of the spinal cord, which is good news for this product. With respect to manufacturing, it's really important you know what you're doing because if you mess this up, your timelines are blown and you waste a lot of money. And again, this isn't our first crack at this. This approach that we've developed based on the iCELLis, we've done it 54 times before. In 15 times, we've done this in a way that supports clinical trials, and we plan to do more of it. So we're working with FAST with a contract manufacturer where our staff go into their laboratory and actually do the work to make sure that we do this well and without delays. So in terms of clinical trial considerations, this is really an important part of the development of a program, and I'll defer to FAST on more specific questions about this if you have those in the panel. But just want to highlight the approach that we're taking. First of all, FAST is going to convene clinician caregiver advisory boards to inform important aspects of the first-in-human study. The first-in-human study will be just some thoughts. It's open label, so you'll know when you're in the study. It's not placebo. A single ICM injection, and that's it. You don't need to go back. There are likely going to be a low dose and then a higher dose, or 2 doses, both of which we believe should be effective based on our animal studies. The primary endpoints in initial study are really to evaluate safety and determining where the product is tolerated. We're probably, the tolerated dose is likely going to be the highest dose because we really don't see dose-limiting toxicities. And the highest dose is probably going to be the most vector that we can give. That's been our experience in other trials. But we will be measuring other readouts of whether this benefits patients. Important part of this is engaging with health authorities to get advice in advance about what preclinical studies additional that we should complete and clinical trial design and manufacturing. They would have seen this manufacturing package 15 times, so there should be no issues around manufacturing. This is the timeline. Where we are now is we've started production of the toxicology lot. We've identified monkeys for the GLP tox study. They are resident to our facilities, so no problem of access there. And we're beginning discussions. We are preparing for discussions with health authorities to nail down the details and working with FAST to secure the contract for the manufacturing facility. And then that would lead to a submission of our regulatory documents and then soon thereafter open up the study. So I'm going to end just on 2 comments, and I'm not one. I'm not a BS person. Let me just put it that way. I say the way it is. I have started 8 biotech companies, and we've worked with those companies in this way. I have partnered with 4 pharmaceutical companies and 3 foundations. We have never seen anything like the engagement with FAST. They are way on the outer side of being truly outstanding. They know what they're doing. In the experience with genetics, you guys are going to leverage to the hilt. There's no drama. There's no ego. Well, there's Allison drama, but, you know, that's... we just call that Allison. But seriously, there's no ego. There's no about we versus them. We've never seen that in any other engagement. And the capability to be able to pull the community together, which is essential here, is probably one of the strongest assets. So I'm gonna leave with my call to charge. We heard from the other speakers, and that is it's time to be disruptive. And what you guys are doing here is disruptive. And you need to be disruptive because those that we usually rely on are not stepping forward. So congratulations on that, and absolutely delighted to be part of this effort, and look forward to further updates as we move the program forward. Thank you.